Yo, what up, class? Welcome to our online lecture this semester. In this lecture, we're going to finish chapter 7, which has taken us forever to finish, but I'd rather us take our time and understand it than rush through it. And then we are going to get into photosynthesis, and we're just going to go through photosynthesis and get as far as we can today and on Monday, and you're only going to be responsible for whatever we cover in class with photosynthesis. All right, so let's get started. So we've basically finished all of cellular respiration, and now we're going to look at which is how you make ATP, right? How the cell makes ATP. It uses cellular respiration, which is this four-step process, and we looked at specifically aerobic cellular respiration, which needs oxygen. So now we're going to look at how do cells make ATP in the absence of oxygen. And there are several organisms that live in anaerobic environments, which means environments that do not have oxygen. So the question is, how do these organisms oxidize glucose if there's no oxygen to oxidize? And so we'll learn that in the absence of oxygen, glucose can still be used as a source of energy using one of two processes, anaerobic respiration or fermentation. So anaerobic respiration, which is without oxygen, it's exactly like aerobic respiration, cellular respiration, except it uses other molecules instead of oxygen as the final electron acceptors at the electron transport chain. And there's some prokaryotes that do this using sulfur, iron, nitrate, sulfates, and metals. And this is typical of organisms that live in deep sea thermal vents and some hot springs and things like this. And then fermentation is simply glycolysis, so the first step of cellular respiration, where you break glucose down into pyruvate. It's glycolysis coupled with a reaction that replenishes NAD+. We'll look at exactly what that means. This does not require the Krebs cycle or the electron transport chain and ATP synthase. Unlike number 1, anaerobic respiration, fermentation does not require those parts of cellular respiration. So fermentation is a little different than classic cellular respiration. So let's recap real quick aerobic cellular respiration. When there's oxygen, most cells make ATP using this process. And the first step, glycolysis, does not require oxygen, but it does need NAD+ in order to break down glucose into pyruvate. So you can see there, this is a figure from your textbook diagramming glycolysis, which you do not need to know at this level of detail. Just look at the beginning. You start with 6-carbon glucose, and at the end, you have 2 3-carbon pyruvates. That's all I expect you to know. Also, I do expect you to know that in the beginning, you use a little bit of ATP to start breaking down glucose. So you can see the first ATP right below glucose you're using 2 ATP. And then at the end, you actually make 4 ATP. So you produce a net of 2. You make 4, but you use 2 at the beginning. So you produce a net of 2. Well, in this process, you can see there in the blue boxes that you need NAD+ in order to break down glucose. So you always need a source of NAD+. The question is, NAD+, when it's used here as an electron acceptor, it's going to become NADH. So how do you get NAD+ in the first place? That's what we're going to look at right here. So here you have glycolysis. Glucose is being broken down into pyruvates. And in order to do this, you need NAD+. NAD+ is going to oxidize glucose. It's going to rip electrons from glucose, and it's going to become NADH. And then pyruvate is going to become oxidized again, and you're actually also going to use NAD+ in this process to make acetyl-CoA, and that's going to go into the Krebs cycle, right? These are the first 3 steps of cellular respiration. So in the process of breaking down glucose, NAD+ is converted into NADH, and then NADH is going to dump its electrons to the electron transport chain, and be converted back into NAD+, so it can be used again for glycolysis. All right, so this diagram here is a variation of how we've typically diagrammed cellular respiration, but hopefully we've gone over that enough to where you understand what's going on. Basically, let me run through it again. So glucose is being broken down into pyruvate, and what you're doing is you're oxidizing glucose with NAD+. It's converted into NADH. Now you need to convert NADH back into NAD+ so you can continue to do glycolysis. If you use up all of your NAD+ and turn it into NADH, you cannot do glycolysis anymore. So you have to figure out a way to make NADH back into NAD+. And the electron transport chain is where this happens. So the NADH made in glycolysis will donate its electrons to the electron transport chain and become NAD+ in the process. It also loses a hydrogen, which is pumped through the electron transport chain across the inner membrane. All right, hopefully you guys are following. Again, essential that you guys look at the other slides, read through the textbook, come to lecture to get what's going on. So NADH donates its electrons to the electron transport chain, becomes NAD+. NAD+ is used again for glycolysis. All right, the electrons from NADH that it donates to the ETC are going to go through the electron transport chain, and finally those electrons are going to be donated to oxygen. So oxygen is the final electron acceptor. That's why you have O2 there. And when oxygen accepts those electrons, it's going to react with hydrogen to form water. All right. Again, I know this is a lot, but this is what we've been covering in class for the last week and a half. Okay. Now I'm going to transition yet again to another diagram. This is good because this is helping you know the material from different angles. All right. So same thing. We're going to start with glycolysis and work through the rest of cellular respiration. So we are recapping aerobic cellular respiration, which is respiration in the presence of oxygen. All right, so in the beginning there, you can see that glucose is being broken down into pyruvate using NAD+. So NAD+ oxidizes glucose and it's converted into NADH. So that's why you see there 2-pyruvate, 2-NADH. In the process of breaking glucose down, you also make 2-ATP. Now the question here is, what happens to NADH produced in glycolysis? Right, because you've made some NADH in glycolysis. And in aerobic cellular respiration, the NADH donates its electrons to the electron transport chain, and it's converted back into NAD+, so it can be used again for glycolysis. This is exactly the same thing we just looked at, except with a different diagram. So here you could draw an arrow at the end of the electron transport chain. NAD+ goes back to glycolysis so that it can oxidize glucose. And then, depending on the final electron acceptor, if it's oxygen, then it's aerobic cellular respiration. Right? The electrons from NADH are donated to the electron transport chain, and those electrons finally are donated to oxygen. And that's cellular respiration. Okay, now let's look at ATP without oxygen. So in the absence of oxygen, some prokaryotes make ATP using anaerobic cellular respiration, which is just like aerobic, except that it uses molecules other than oxygen as a final electron acceptor. So here, the electrons donated by NADH to the ETC end up being donated to nitrates, sulfates, carbon dioxide, iron, and other things. All right, and then there's one more way of making ATP without oxygen, and that's fermentation. So fermentation is simply glycolysis, which is this first step on the diagram, coupled with a reaction that replenishes NAD+, so that the cell can continue to carry out glycolysis, right? And that's it. Fermentation simply is glycolysis, and then it has to figure out another way to generate NAD+. And the way it does this, if you read there, it's that the electrons from NADH reduce pyruvate, and in the process it makes NAD+. We're going to look at that a little more closely in a second. One thing I want to mention though is that the amount of ATP that can be produced varies from in each of these three ways of making ATP. So aerobic respiration, so here you can see in the bottom a little gradient from high to low of ATP. And it says additional ATP produced because you've made 2 ATP in glycolysis. So aerobic respiration generates the most ATP, and it's 32 ATP per glucose. That's including glycolysis. Then anaerobic respiration also makes quite a bit of ATP, but not as much as aerobic. But finally, fermentation makes the least glucose. It only makes 2 ATP per glucose, and that the 2 ATP come from glycolysis. All right, so remember, fermentation does not do Krebs cycle, it does not do electron transport chain, it just does glycolysis and then figures out a way to replenish NAD+. So let's look at fermentation super quickly. Again, fermentation uses glycolysis to make ATP and then reduces, so how it replenishes NAD+ is it reduces pyruvate using NADH. So in muscle cells, this happens in muscle cells when they run out of oxygen. In muscle cells, after glycolysis, they reduce pyruvate into lactate or lactic acid. Same thing, basically. But they do this using NADH. So you can see there in the diagram, glycolysis, NAD+ oxidizes glucose into pyruvate, and you make NADH in the process. That NADH is then used to reduce pyruvate into lactate, to convert pyruvate into lactate. And as NADH is used to convert pyruvate into lactate, it becomes NAD+, that can then be used again for glycolysis, right? So it's a little cycle. This is a way that cells have figured out to replenish NAD+. That's really what fermentation is. This also happens in yeast and other microbes. Well, let me back up 1 second. So in muscle cells, the reason this happens, normally muscle cells will make ATP using cellular respiration. But say you're doing exercise and you're doing so much exercise that you're making so much ATP that you run out of oxygen, right? Which is why you begin breathing heavy, because your body needs desperately more oxygen. But say you've run out of oxygen in your muscles, so then your muscles begin making ATP using fermentation. All right, and then in yeast and other microbes, they also can do glycolysis without oxygen. Or I guess I should say they can make ATP without oxygen by using glycolysis and then reducing pyruvate. So they reduce pyruvate into ethanol, and it releases CO2 in the process. And this form of fermentation is what's used to make beer, wine, bread, cheese, basically all of the good things under the heavens and on this earth, right? So you can see here, again, glycolysis converts, or NAD+ is converted into NADH during glycolysis in order to oxidize glucose. And then pyruvate is actually going to go through a step where it loses carbon, one of the carbons as carbon dioxide, and then acetaldehyde is reduced to ethanol using NADH. Same concept, you're just wanting to replenish NAD+ levels. So you are using NADH to reduce pyruvate, acetaldehyde in this case, and make NAD+ in the process. All right, we can go over this again in class if it's not clear, but we'll move on for time's sake so this online lecture doesn't run forever. Okay, so what is the main advantage of fermentation over aerobic cellular respiration? A, fermentation generates ATP even if oxygen is not present. B, fermentation generates more ATP per glucose than aerobic cellular respiration. C, fermentation does not generate toxic byproducts such as CO2. And D, fermentation gets rid of pyruvate, which would otherwise accumulate in the cell. I'll let you guys think about it real quick. And the answer is A, right? Fermentation, the advantage of fermentation is that it can generate ATP even if there's no oxygen. So when your muscle cells run out of oxygen, boom, it can use fermentation. And that is the advantage it has over cellular respiration. Alrighty, now let's transition into chapter 8, photosynthesis, finally. And we'll cover a significant chunk today, and then we'll see. We're definitely not going to get through all of chapter 8, so we'll cover a little bit on Monday. Okay, so what is photosynthesis? It's basically turning light into sugar. It is, you throw in light on one end of the machine and you get sugar out of the other end. It is like something out of the Willy Wonka book or movie, whichever you prefer. But technically what it does is it harvests energy and light, kinetic energy, right? And converts it into energy and glucose, potential energy. So it's an energy conversion. Really, that's all that photosynthesis is. And it uses CO2 as a carbon source for glucose. So really you have light and carbon dioxide as the starting materials, and then sugar is the product. Sugar is the main organic product of photosynthesis. It is the whole point of photosynthesis, is to make glucose. And why do they care about glucose? Because it is an energy source for ATP production, just like it is for us animals. It is the same thing. Plants need glucose in order to do cellular respiration and make ATP, right? Remember, that was a big deal that we went over in the beginning of chapter 7, the previous chapter, is that both plants and animals do cellular respiration. So what the difference is that plants make their own food, make their own glucose in order to do cellular respiration, whereas we have to consume it, right? So yeah, the main reason, let's see, what am I trying to say here? The main reason to make ATP is as an energy source for ATP production. And then the other reason they make ATP is as a building block to make cellulose. So remember, plant cells have a cell wall that is really tough, and it's made out of cellulose. So in order to continue to grow, plants need to make more cellulose, which is really made out of glucose. Cellulose is just a polysaccharide of glucose. So plants use simple inorganic ingredients to make complex organic food. Another way of describing photosynthesis. But they use carbon dioxide and water along with energy from sunlight. And they make sugar, glucose, and oxygen. Those glucose being the main product, and oxygen is just a byproduct. They don't care about making oxygen. It's just a byproduct of the whole photosynthetic reaction. And something really handy is that the formulas for aerobic respiration and photosynthesis are simply the reverse of each other. So these are actually very related, and we'll see more on this later. But aerobic respiration, which is what we just covered in chapter 7, you know that it breaks down glucose in order to make ATP. So you start with glucose, which is a C6H12O6, and use oxygen as a final electron acceptor. You release CO2 as you strip off every carbon from glucose. You produce water as a byproduct, and you make ATP, which is potential energy. So here the energy conversion is potential energy in glucose is converted into potential energy in ATP. Now in photosynthesis, plants are taking in carbon dioxide from the atmosphere, water from the roots, and using sunlight as a source of energy in order to make glucose. So sunlight is kinetic energy. And this makes glucose and releases oxygen as a byproduct. So you'll see though that they're the reverse of each other. They occur in opposite directions. So if you learn one, you'll know the other. My tip. And for this test, you definitely need to know these formulas. You don't necessarily need to know how much glucose, how much oxygen. Just need to know that the word formula, right? What the starting materials are and what the end products are. So why is photosynthesis important? Photosynthesis is essential to life because it is the point at which energy enters living systems and carbon enters living systems. So here you have the little energy cycle that we looked at in the beginning of chapter 6. And you have kinetic energy from the sun that enters our atmosphere, our ecosystem. It's used by plants to make glucose using photosynthesis. And then that glucose, that sugar, is eaten by other animals, which use it for cellular respiration to make ATP. And it releases CO2 and hydrogen and water in the process. And that CO2 and water is actually used by plants to do photosynthesis and make glucose. So photosynthesis there, you can see that it is where energy enters living systems. So all living things depend on plants because they harness energy from the sun and turn it into usable energy, into sugar, right? And also it's where carbon enters living systems. This is important because remember, all living things are organic matter. They're composed of organic matter, which is mainly made of carbon, hydrogen, oxygen, nitrogen. Carbon being the main atom in all of this. And so carbon enters living systems, plants and animals and everything, because of photosynthesis. So photosynthesis uses carbon dioxide, which you see there, as a source of carbon for sugar. So if it wasn't for plants, we wouldn't be able to, living things wouldn't have a source of carbon. Because here's the big deal. Plants can take an inorganic form of carbon and turn it into an organic form that can be consumed by other living things. So plants are the only living thing that can take an inorganic form of carbon, carbon dioxide, and turn it into organic carbon in the form of glucose. We cannot use carbon dioxide as a source of carbon for building things. Plants can. So plants are the point at which carbon enters living systems. All right, quick question. Evolution favored the development of photosynthesis because photosynthetic organisms, they A, produce oxygen, B, make their own food, C, make food for heterotrophs, which we already looked at. Heterotrophs are organisms that eat other organisms. D, use carbon dioxide, or E, all the choices are correct. So which of these reasons are the reasons that evolution favored the development of photosynthesis? Okay, so B is the right answer. Photosynthesis allows plants to make their own food, and that is evolutionarily very advantageous if you don't have to wait and consume other organisms for food, right? A is wrong because plants don't care about producing oxygen. Oxygen is no advantage to plants. I mean, I guess it is ultimately, but the main thing is that they can make their own food. C, plants didn't care about becoming food for heterotrophs. They actually wouldn't want to become food for heterotrophs because then heterotrophs, the herbivores, would kill plants, and that would not be good. And D, they use carbon dioxide. Again, there's no advantage to using carbon dioxide. The earth wouldn't, or the plants didn't care that carbon dioxide is a poisonous gas. Plants just were using it as a source of carbon. So yeah, the main evolutionary advantage was B, that they could make their own food. Okay, so let's do a little overview of photosynthesis and look at first where photosynthesis occurs. And by now we all should know that photosynthesis occurs in the chloroplast. So if you take a leaf and you do a cross section, you'll see that it has multiple layers of cells. In that middle layer, called the mesophyll, you have cells, plant cells, that have chloroplasts, which are those green organelles in the diagram. And if we zoom into a chloroplast, you can see that a chloroplast has an outer membrane, an inner membrane, and then inside it, it has internal membranes called thylakoid membranes. And these membranes contain chlorophyll and other photosynthetic pigments that cluster into these structures called photosystems. And we'll look at what those are in a little bit. So they're the thylakoid membranes. So these are really the important membranes in chloroplasts. Now, what's interesting is that chloroplasts are similar to mitochondria in that they have internal membranes, these thylakoid membranes, and the inner membrane as well. They have their own DNA, circular DNA, and their own ribosomes, which are different than the ribosomes of the plant cells these chloroplasts are found in. So this is just like mitochondria, and these features resemble features of bacteria. It's almost like they are bacteria or once were bacteria. So chloroplasts are thought to have arisen from endosymbiosis, which is the scientific theory that suggests that ancient cells once engulfed other cells that then became the mitochondria, or became the chloroplasts. And this becomes clear too when you compare chloroplasts on the left with cyanobacteria on the right. And cyanobacteria are prokaryotes. They're free living, so we actually looked at some under a microscope. They form those filaments, and they have an inner membrane. Really, they have folds of membranes, so they don't technically have inner membranes, but they do have their own DNA, their own ribosomes, and these cyanobacteria can do photosynthesis. They have the machinery to do photosynthesis. So perhaps chloroplasts came from ancient bacteria that resembled cyanobacteria. Okay, so now let's look at the actual process of photosynthesis. And we're going to break down photosynthesis into two steps. The first step are the light reactions. And this is the photo part of photosynthesis. Photo means light, right? Photograph and photo whatever. Just photo, the root word there means light. And so what photosynthesis does, or what the photo part, what the light reactions do, is they take the energy from sunlight, which is kinetic energy, because the light is made of photons that move. They use that energy to produce ATP and NADPH. So NADPH is just a chemical cousin of NADH. It is an electron carrier, and that's all you need to know. And in this process of taking the light energy and converting that into energy in ATP and NADPH, you release oxygen as a byproduct. So oxygen, again, plants are not interested primarily in making oxygen through this process. They're interested in making glucose. Oxygen is just a byproduct. And then you have the carbon reactions, which are the synthesis step of photosynthesis. So this is where we're actually going to synthesize sugar or glucose. So these reactions use the ATP and the NADPH made in the previous step to synthesize glucose made using CO2 as a source of carbon. All right, so that's kind of the breakdown of this little diagram. If we zoom in and take a closer look at this, so now we're actually looking at where photosynthesis happens, and this is in the thylakoid membrane. So again, these are those little squiggly lines in the chloroplasts, right? So the first step is the light-dependent reactions that make the ATP and the NADPH. So they occur in the thylakoid membrane, and it's cool, it's funny because these reactions consist of an electron transport chain that powers ATP synthase. Very similar to cellular respiration, except that it gets its electrons from photosystems. All right, and we'll look at exactly how that happens. But if you look at the diagram here, the energy in sunlight is hitting the photosystem, which is actually going to release an electron there in the black arrow. And those are going to power the electron transport chain, which is going to make ATP, and it's going to release NADPH in the process. And then the next step are the carbon reactions, which is also called the Calvin cycle. And so these reactions happen in the stroma, which is just the liquid inside of the chloroplast, the fluid that the thylakoid membranes are floating in. So these reactions are the money maker of photosynthesis. These are the ones that are making the glucose. So they're going to use the ATP and the NADPH made in the previous step to power the synthesis of glucose out of CO2 and a 5-carbon molecule that's cycling in this Calvin cycle. And we'll break these two steps down and look at them closely. So first, the light-dependent reaction. So in order to understand this, we have to understand light. And light is made of high-energy particles called photons. So we looked at this when we were looking at the lab that looked at absorbance. But again, light has these little photons that travel in waves. And photons have different wavelengths. Shorter wavelengths have more energy. Longer wavelengths have less energy. So if you look at the spectrum here in this diagram, on the left is the shortest wavelength, on the right is the longest wavelength. And you can see that in the middle there is a visible light spectrum. So the light we can actually see is a very small part of the whole light spectrum. In that light spectrum, starting from left to right, you have gamma rays, X-rays, UV light, which is the light that burns us from the sun. You have the visible light spectrum, which are the colors we see. But then you have infrared, which is what Marines see using their night vision goggles. You have radio waves, which is what powers the car, not powers, but it's what carries the radio stations that we hear on our car. So all of that is a light spectrum. We can only see that tiny, tiny little portion called the visible light spectrum. And that's where the energy in light that plants capture is found in. So the way they do this is plants have pigments that capture these photons. Chlorophyll A, chlorophyll B, and carotenoids are the most common pigments in plants. And most of these absorb high energy blue light and red light. So you'll see there in the diagram the visible light spectrum and 3 different pigments there, the 3 that we mentioned. And you can see that most of them capture energy in the violet blue spectrum, end of the spectrum. And then some of them also catch a little bit of the red spectrum of light. And the colors that are not absorbed are reflected, right? So we looked at this again when in that lab looking at absorbance. So the photosynthetic pigments are absorbing the colors that are under those peaks. And so you can see that the green and yellow spectrum is not absorbed really well. And so this is why most plants are green, because they're reflecting green. Right? They're absorbing violet, blue, red, a little bit of yellow, and then reflecting green. And so that's why plants are green. And why do leaves turn color in the fall? Well, when temperatures drop, the first pigments that start breaking down are chlorophyll A and B. And so as chlorophyll dies, the plants start reflecting reds and oranges, which are at the end there. And so plants begin reflecting those colors and they turn really bright red, really bright orange. I'm in Ann Arbor right now and the trees are on fire. It's crazy, super cool. And the reason the trees are bright red is because temperatures have dropped, chlorophyll A and B have died, carotenoids are still around, and they reflect orange and red. Now, one more thing I'll mention is that chlorophyll, both A and B, they absorb more in the shorter spectrum of wavelength, especially chlorophyll A. It absorbs more in the violet. And so chlorophyll is able to absorb the most energy out of all of the pigments. And also chlorophyll can absorb in the red spectrum, right? Which is why it doesn't reflect red. But so chlorophyll can absorb a lot of light. Okay, so cool little question. This is an actual experiment that we could do in the lab if we wanted to. But in this experiment, light has been split into its colored spectrum. So we use a prism, and that prism splits white light into the different colors. And you put a filament of green algae under that spectrum. And then you add oxygen-sensing bacteria, so bacteria that love oxygen, that move to to areas of more oxygen. So you tell me what wavelengths of light produce the most oxygen. And you can see here that the violet blue wavelengths and the orange red wavelengths produce more oxygen, and you see a dip there of the light absorption. So just to explain, on the y-axis you have light absorption from low on the bottom to high on top, and so the peaks are on the blue violet part of this end of the spectrum and on the orange red end of the spectrum. And then the green yellow, you see a dip because the photosynthetic pigments don't really absorb those colors, they reflect them, which is why, again, leaves are green. All right, so let's look at the light reactions and how they're harvesting this light energy. So chlorophyll A, which is the most efficient pigment, and the other pigments reside in structures called photosystems, which are simply a matrix of proteins and pigments that cluster together. And as these absorb light, which are photons, the photons excite the chlorophyll, which begin a series of energy transfers from one pigment molecule to the next. And this isn't really understood how this energy is transferred. So again, there's still a lot to learn in biology, but we have learned a lot. So we do know that the photons hit the chlorophyll molecules, they excite each other. So that's why you see a little black line jumping from chlorophyll to chlorophyll, even carotenoid to carotenoid. So there's an energy transfer there. It's thought to be kinetic energy. So it's thought that the photons excite chlorophyll. The chlorophyll actually changes structure, and these structural changes excite the next chlorophylls that change structure, and you just have like little vibrations that are being transmitted. I think, as far as I can remember, that's what's going on. But finally, this energy reaches a reaction center chlorophyll, which is chlorophyll A, if I'm not mistaken. And in turn, this chlorophyll is going to release electrons. So basically, the photons begin a chain reaction of energy transfers in the photosystem, culminating in the release of 2 electrons from this reaction center chlorophyll. And these 2 electrons are then going to go to the electron transport chain and power some hydrogen pumps, just like in cellular respiration. All right, so this photosystem is on the left that we just looked at. The photons excite the electrons, or sorry, the pigments, and then these are going to release 2 electrons that are going to go through the electron transport chain. And you can see there that there's a hydrogen pump that's being powered to pump hydrogen ions through across the membrane. And as these hydrogen ions accumulate inside the thylakoid space, they're going to create a concentration gradient, which is going to drive their movement through ATP synthase to generate ATP. So honestly, very similar to cellular respiration in this aspect. Now, in the photosynthetic electron transport chain, NAD plus is a final electron acceptor, not oxygen. So if you look there, if you follow the black line of the electrons through the electron transport chain, you can see that they will eventually end up converting NADP plus into NADPH. All right, so this is where NADPH is made, by reducing it from NAD plus to NADPH. And then ATP is made by synthase. So now you've made ATP and NADPH. Now remember, the photosystem, the first one, has released electrons, right? Basically, the sunlight has excited the photosystem, releases 2 electrons. Now those electrons need to be replaced. And in order to replace those lost electrons, water donates 2 electrons to the photosystem and is split into 4 hydrogen ions and oxygen. So this is where oxygen is released. Okay, so to kind of summarize the light reactions, let's run through this little slide again, and I'm going to try and point out important parts. So you start with the photon on the left, right? The light excites the photosystem and it releases 2 electrons. Those 2 electrons go through the electron transport chain, powering hydrogen, this hydrogen pump. It's going to pump hydrogens across the membrane, and you're going to end up making a concentration gradient. So you're going to have high concentration of hydrogen in the thylakoid space. Those hydrogen ions are going to move through ATP synthase because of the second law of thermodynamics, basically diffusion, going from high to low. and that's going to power ATP synthase to make ATP. So this is where you make ATP. And then the electrons that have been going through the electron transport chain are finally going to be donated to NAD plus to make NADPH. And this is where you make NADPH. So the two main products of the light reactions are these two here, ATP and NADPH, because they are the energy source for the carbon reactions that follow. Now, you've lost 2 electrons in the photosystem, so you have to replace them. Basically, a general rule in chemistry and biology is if you lose it, you got to replace it. So those 2 electrons are going to be replaced by water. Water is going to donate 2 electrons to the photosystem, and in the process, it's going to be split into oxygen and hydrogen ions. So this is where oxygen is made and is released into the atmosphere. And that is the light reactions. And on Monday, we're going to look at the carbon reactions, the Kelvin cycle, which is really short. That's all we're going to look at. We're also going to look at how photosynthesis and cell respiration are tied together, and we'll end there. We're not going to look through the last sections of Chapter 8. So I'll be assigning homework on Chapter 8 on Monday, and then, we'll do a review. On Monday, I'll be moving the study group from Monday after class to Wednesday after class. I know that might be inconvenient for a few of you, but it's the only time I have. Monday. My Monday got booked by another obligation at Richland, development program, professional development program. But anyway, hopefully you enjoyed our online lecture. If you like these, let me know, and maybe we can do more of these instead of coming to class. I don't know. I mean, we'll for sure still need the classroom to answer questions and stuff, but maybe we can do these, and in class we can answer questions. So if you like these, let me know, and I will do more of them. All right, so see you guys on Monday. Enjoy the weekend. Hope you watch this lecture as many times as you want. It'll be on YouTube until the end of the semester. All right, adios, guys. Bye.